Hello everyone, welcome to the penultimate thesis talk of this term. The talk will be about an hour long and then there will be a Q&A session. Both the talk and the Q&A session will be live streamed on YouTube and also recorded. Now, please let me introduce our today's speaker, Dr. Freya Jephcott. Dr. Freya Jephcott is a junior research fellow at Queen's College, University of Cambridge, and a member of the Cambridge Disease Dynamics Unit. Her research focuses on epidemic outbreaks and public health. She's also a consultant for the World Health Organization and works for the Medicine Sans Frontières as a manager of epidemiological activities. In today's talk, Dr. Jephcott will be speaking about the outbreak investigations of newly emerging infectious diseases. Please welcome Dr. Jephcott. Dr. Jephcott, the stage is yours.
Thank you for that. I'm so excited about giving this talk today. I love the Trinity College Science Society talks, so this is a very big privilege to me. I'm just going to start sharing screen, so please do let me know if this doesn't work. Oh no, it says the host has disabled screen sharing.
Now it should be possible.
Let me work out.
Great.
Perfect. Okay, so now I'll begin in earnest. I think, as was already said, my main area of interest is the efficacy of outbreak surveillance and response systems. So that's really how well our formal systems for detecting burdens of disease within communities actually work, and if, which hopefully they do, how good they are at responding to these burdens of disease and sort of meaningfully bringing them down under control. I will try and shift this a little. There we go. So one of my favorite parts of the Trinity College Science Society talks is when you get a bit of a sort of biography of the speaker. So very quickly, I originally studied medical science back in Australia at the ANU. I focused on virology and epidemiology. And initially the plan was to graduate medicine and become a medical epi, but I took what I thought was going to be a one-year detour to do some combined sociological and epidemiological research. But that actually ended up turning into a master's, bringing together epidemiology and anthropology, and then a PhD, and then a research fellowship. And actually, just to update my affiliation, I'm no longer a junior research fellow with Queen's College. I'm actually a research associate with the Centre for the Study of Existential Risk, but still working in roughly the same area. Now, one of the reasons why I didn't end up going back onto my original career trajectory was that I found that the social sciences gave something to my work trying to understand outbreaks that it was just too hard to give up. The thing that they gave me was the ability to look at outbreak responses as they were unfolding on the ground in all of their sort of complexity and messiness and all of the unintended consequences, and to consider all the kind of factors that were informing that, even the things that aren't easily quantified. So to put that in sort of more hard terms, what it has allowed me to do is make detailed case studies of outbreaks, either in real time, so that means me watching outbreak responders as they sort of grapple with an outbreak and taking very detailed notes, or sometimes these sort of retrospective ones where I just interview everyone involved, or I gather up all of their documents from the time, and I mean everything, like lab reports, email chains, WhatsApp messages if they still exist, everything, can sort of try and construct a detailed account of the outbreak that way to analyze. Now, as has been mentioned, in addition to my research, I do a bit of applied work with organizations like Médecins Sans Frontières, so intermittently managing outbreaks. Some of these are complex outbreaks in resource-limited settings where it isn't clear what the etiology is, but most of the time it's Ebola. And I also work with the WHO. So it started out doing event verification, then something called preparedness missions, going to countries that are susceptible to an outbreak spreading into them and seeing if they're ready for it. And it slowly turned into me doing more policy work, especially around guidance and tools for managing outbreaks of unknown origin. So today's talk, I thought I would start by looking at the processes of hypothesis building that can occur and sometimes go wrong during outbreak investigations. And then I would look at a few other features of outbreak investigations and responses, namely developing case definitions, laboratory testing, and maybe some of the doctor-patient relationship work. And those last three I'll talk to in relation to COVID. And this first bit, this hypothesis building, I'll talk to in relation to a case study I developed from Ghana in about 2014, which I'm particularly fond of. So I realize I'm sort of incentivizing some people to get through the Ghana talk to get to COVID, but sorry. All right. So, sorry, that's a bit of an overview. The green bits are outbreaks where I've done research on this. The blue bits are outbreak responses I've participated with as a responder. Just to make it clear, I'm not throwing shade at outbreak responders, I'm there too. And the red bits are retrospective case studies I've developed. And I'll try and incorporate some of them into the second half of the talk. So, hypothesis building during outbreak investigations. I think a bit of background. First, emerging infectious diseases. Even before COVID, they were kind of a big deal in public health. In fact, since maybe the late 1980s, they've been a sort of big part of the public health agenda because of the realization that HIV had somehow emerged, spread around the world, and was causing a major public health issue in the form of AIDS. But also around that time, we noticed that a lot of other infectious diseases were emerging, and some that had been thought to kind of be going away or at least under control started increasing in frequency and size too. So that led to a lot of investment in understanding emerging infectious diseases, in particular research in microbiological, some mathematical modeling and such, and then also a lot of investment in technological development in the forms of clinical treatments, diagnostic tests, vaccines, surveillance technology. Something that initially, in particular, has been neglected, though, is trying to reflect on the core processes of responding to emerging infectious disease threats. These core processes include the sort of way we investigate outbreaks and work out what's going on the ground. This image that I'm hoping you're all seeing right now, that's meant to be John Snow trying to explain that he thinks that cholera is coming from a water pump to a group of skeptical, I assume, public health officials. The way that he kind of came to those conclusions about what was causing this large cholera outbreak is more or less how we do it today. This sort of process of investigating outbreaks has essentially been the same since the 1850s. There are two reasons, I think, why we haven't really reflected more on our processes of investigating outbreaks. One of them is the way we tend to think about this process. We tend to think about this process either as a series of steps, which if you follow closely will inevitably lead to you identifying the correct causative agent and being able to develop some kind of meaningful intervention that helps with the burden of disease you've uncovered and characterized. It also suggests the way that we often present these steps of investigation, that if you're unable to work out what's gone wrong or the agent that's caused it, it's because you in some way didn't fulfill one of these steps properly. The other way you often see outbreak investigations being characterized is that they were all the work of one like absolutely profound genius individual, typically a white person in a lab coat that somehow has single-handedly done all of the steps. You'll see that a lot in biographies of epidemiologists, especially Western ones. So that picture I've stolen there is from a recent CDC biography of outbreak investigations. And you can see that I'm not exaggerating when I say this idea of profound individual. But that characterization doesn't really open itself up to much academic inquiry, because saying that it's the work of the little gray cells of some genius is not very accessible to people like me. So these two kind of characterizations don't really give us much to work on. And it's hard to know whether they are sort of accurate depictions of what's going on the ground. This is because of the second problem, which is the way that we tend to publish in epidemiology and applied epidemiology, which is in a sort of smooth depictions of what happened on the ground, things that don't really allow for a lot of the uncertainty in the sort of journal articles. You don't really say, So I sent the samples, and supposedly they were run on this test, but I'm pretty sure that they left them in the mailroom for 2 days and that they defrosted, and I don't really trust that guy to run the tests anyway, and all of that kind of messy stuff that leads to a lot of uncertainty. The other issue is that we tend not to publish when we're not really sure what happened there. Which happens a lot. So it ends up giving us a sort of sample bias, with most of the times we publish the investigations where we ended up being fairly certain that we worked out the causative agent that was responsible and more or less characterized why it suddenly emerged or why it spread through a population. And that kind of, those cases tend not to be really the interesting ones to look at anyway. Okay, so in talking about hypothesis building, like I said, I'm going to look at one particular case study in great detail. And processes of hypothesis building, they're a lot more complex and a lot more subject to all these kinds of influences than I really have time to go into here. So I'm going to focus in particular on the way that I guess certain sort of constellations of professionals who come together for emerging infectious disease outbreaks or any kind of high profile outbreak in a resource limited setting, so perhaps a low middle income country, affect hypothesis building. So it's kind of, I've made it a little narrower there. With emerging infectious disease outbreaks, they do get a lot of attention, even if they're just sort of suspected emerging infectious disease outbreaks. And with that attention, they get slightly different from if it was your kind of an area that normally gets cholera and therefore has kind of cholera under control, and it's not that exciting to hear that this area has cholera, terrible as it might be on the ground. Now, when people think they have a suspected emerging infectious disease outbreak in these settings, what tends to happen is the regular local public health officials, so that's the district disease control officers and the clinicians and such, they get joined in the investigation by, say, national health authorities, or sometimes NGO groups like Médecins Sans Frontières, or international health authorities on occasion, like the World Health Organization, our research groups, and sometimes even private industry. Now, in theory, that shouldn't change too much, because if you go by, say, the normal guidance for investigating an outbreak, so for somewhere like Ghana, that would be the Integrated Disease Surveillance and Response protocols. They would say that all of these additional characters, they're just supplementing the knowledge and resources of those local public health officials who will stay responsible for the investigation and seeing that this public health emergency is brought under control. What actually happens on the ground is a bit different though. All right, so I'm going to very quickly go through the methods, and I'm going to do my best to make it as clear as possible, though I realize I've just thrown a lot of rainbow dots at everybody. To make this particular case study of this outbreak I'm about to describe to you, I collected all the primary documents I could from the time. So every email chain, every text message, every note that somebody scrolled on the back of a test tube genuinely about the case. And I compiled them all into a sort of timeline of information. I also interviewed everybody that participated in the response, and I was actually there for it, for most of it myself. So I got to watch them in real time and what they were doing and talk out kind of the activities that were being undertaken. Now, from that, sorry, I'm having mouse issues. I was able to develop this rainbow monstrosity, which shows the spread of information across the outbreak investigation. This is a bit of a weird one, so bear with me. Each of the different colors represents a particular professional that was involved in investigating this outbreak. So the main pediatrician in the outbreak is this certain green color, and an American researcher, a naval researcher, is a nice yellow color. And then these lines between them, that just sort of follows them through the response. But where a color, a colored circle touches another colored circle, that's where some information was exchanged. So where there's, say, a certain kind of reddy pink, which is a virologist, next to the green that was the pediatrician, that's either them having a phone call or sending some emails or passing a document that shared some information about this outbreak. So this is really a depiction of how a narrative of an outbreak was built. Okay. All right, I'm now going to do my best to run through what was a painfully complex outbreak in like the space of 5 minutes. So bear with me. So 21st of November, maybe 2010 to the 17th of December, 2011. So a space of a few weeks, this little mission hospital in a city called Techiman, I received 16 children with signs of, well, they came in with sort of seizures and paralysis and sort of varying states of consciousness. Now, these children came from around this area called the Brong-Ahafo region of Ghana. So Ghana is in West Africa. It's where that little red dot is on the map of Africa in the corner. And the Brong-Ahafo region is pretty much in the middle of Ghana, so sort of halfway up and in the center. So what happened when these children presented to this mission hospital was the house officers at the time took a look at them and understandably were like, well, this is probably cerebral malaria. So they tried treating them for malaria, but none of the children improved. So they went and grabbed a German pediatrician who was working at the hospital to try and get a better sense of what might have been happening. Her first instinct was that this was probably viral encephalitis, looking at the children, but she wasn't sure what could be causing it. So in line with the kind of national protocols for this sort of situation, she summoned someone called a district disease control officer, so one of those local public health officials. The public health official came, took a look and was like, you know what, I reckon they're probably just meningococcal meningitis or cerebral malaria. Don't worry about it. The pediatrician was not really contented with this analysis because they didn't look quite right and they weren't responding to treatment. So a second district officer was kind of called up and pulled in on New Year's Eve, actually, took a look at the kids, thought that the pediatrician was making a pretty compelling case and called an associate of his who happened to be working in a specialist infectious diseases laboratory in the capital, Accra, a virologist. He talked to the virologist thought he could do something. So the district officer grabbed samples from some children, got on an overnight bus all the way down to Accra. At best, it takes maybe 6-7 hours. And on New Year's Day, gave him this cooler of samples. The virologist ran the samples on some tests and ended up getting a strange result, which was the test suggested that it was something called B virus or monkey B virus. Now, this virus is a simian herpes virus. It's typically associated with a fatal encephalitis in East Asia. So with this kind of giant announcement of this lab result, essentially all the sort of national heads were notified. So the head of the Ghana Health Service, the head of the Ministry of Health, other members of the field epidemiology team. Now, the field epidemiology training program is the specialist program in Ghana, which trains doctors and vets and people with backgrounds in sort of bench sciences in applied epidemiology. And as part of this 2-year fellowship, they can also get some on-the-ground experience investigating outbreaks. So with this sort of bombshell that this was B virus, which I should say had never been described before in Africa, they sent one of these field epidemiology training program teams out to investigate. They got to the Brong-Ahafo region, they did their investigation, they came back down to the capital and they gave a big presentation to the heads of the health service and such, and said, look, what we think's happening is that whilst parents are away on their farms, they're leaving their children unattended, and these children are interacting with monkeys and contracting this terrible virus. They also suggested that the reason why these cases were coming from quite a spread out geographic area in the Brong-Ahafo region, because they weren't sort of clustered together, it was sort of one child from this village, one child from another, was that there was this forest belt that connected all of the affected villages and that the monkeys lived in this forest belt. So, I mean, that kind of came together quite nicely, and it did end up as a sort of small little paragraph in a kind of publication. But the thing is that if you start to look at the epidemiological data that sort of fed into that picture, that sort of amalgamated account, it doesn't really hold water. It begins to get a little sketchy a bit quick. Tends to get a bit sketchy a bit quickly. So I should just point out that the reason this talk was rescheduled is because I currently have something horrible called long COVID, which means I got COVID in April and I'm still kind of reeling from it now. And one of the maybe lesser known side effects of this condition is struggling with language. So if I say something completely daft, please let me know, but also bear with me if I'm a little all over the place. Okay, so back to scrutinizing this epidemiological data. When I started to go into these documents and talk to people, it became clear that the clinical picture wasn't as indicative of viral encephalitis as might have seemed from people telling me about this outbreak. A lot of the children, if not most of them, were afebrile when they came in, which is a bit unusual for an infectious disease. Also, the onset of the symptoms had been quite sudden. Some of the children had been dancing, one had been playing football, another had been talking to their parent, and they just collapsed onto the ground, seizing or paralyzed with sort of epistatonus. Another thing was a lot of the children had normal white blood cell counts, also a bit unusual for an infectious disease outbreak. So already it seemed a little odd. Now, the next thing that kind of came up was the test results. So there had been that laboratory test result that said this was B virus, this very rare, never before seen in Africa, simian herpes virus. Well, it turns out that the test that gave up that result was this unvalidated experimental, what's called a multiplex PCR platform that some Japanese scientists had put together for East Asia and could in theory test for about 156 different viruses known to cause a sort of encephalitis in humans. But it had been kicking around that lab for years and years and years. It had been defrosted many times, and it was a bit worse for wear. And there weren't actually the resources to run controls on that test at the time either. So suddenly that like laboratory confirmation becomes a lot shakier too. Now, actually, although it wasn't captured in the original story, later on some samples were actually sent to a simian herpes reference laboratory in Georgia. And when, again, it's a little unclear, but when they ran the test, they said that whilst the samples they had were negative for B virus, there'd maybe been something positive for what's called herpes papio 2. Now, herpes papio 2 is found in baboons. It's never been described in humans. It's not clear that it has zoonotic potential. And actually, when I talked to people about this, no one could quite remember what the results really were. And also the two samples they used were from much later cases and had actually tested negative for B virus on the original platform. So that again becomes quite shaky. Now, I guess what I thought was maybe the smoking gun and maybe why I started looking at this investigation in the first place is because I happened to already be living where this was all unfolding. And there wasn't a forest belt and there weren't wild monkeys, which for a whole outbreak that's sort of predicated on being spread by wild monkeys within a forest belt seemed like a kind of key missing ingredient. This is a Google Maps picture of the area, and those dark green bits towards the south are dense forest. And as you can see, most of the area is quite plain. It's a sort of grassland savanna area. And the numbers, those little circles, they represent villages where there were cases. And if you look to the right, there's some, I think it was made in Microsoft Word, a sort of a map that the district officer made me that supposedly shows this forest belt that connects all these villages. Well, I've tried to turn that into a red line on top of the Google map. And I hope, as you can see, there just isn't a forest belt. Now, that's not to say that there aren't any monkeys in vicinity. There's a monkey sanctuary that's technically in the region, but not near any of these cases. And there was a potass monkey found during the field epidemiology team's visit, and it was decapitated. It was someone's pet, and they tested it, but it was negative for B virus. So one monkey was found and killed, and it was negative, and there is no forest, and there's not really wild monkeys running around. Further, I should say, there just aren't macaque monkeys in sub-Saharan Africa, the animal that's known to carry B virus. It's found in East Asia, which is that map to the top right of the screen. All right, so, also I should say the field epidemiology team's investigation only took about 3 days, and they only spoke to 3 affected families of the 16 children. So it all gets a little thinner and thinner. Now, none of this is to say that there wasn't an outbreak at that time, and it wasn't caused by this never before seen herpes simian virus in this group of children. It's just to say that it's unlikely. And actually the unlikeliness of that theory isn't an issue either, because actually when you're investigating an outbreak, creative leaps are often necessary to begin to work out what's going on. Where we hit the problem is that this quite exciting theory, and this investigation and response went on for years actually, was able to kind of continue despite being quite incongruent with a lot of the evidence. It was able to continue whilst other more plausible explanations were never actually investigated. So more plausible ones might be a toxic etiology, which might fit better with a lot of the kids being afebrile, and the epistotonus, which if you don't know what that is, it's where you sort of, the spine curves back on itself. I think it's normally associated with tetanus, but I think of it as sort of the Archaeopteryx fossils. Anyway, it's often a kind of more indicative of a toxic etiology. So I guess the question then becomes, just for the books, all of the people involved with this I know very well. They're really talented virologists, infectious diseases researchers, physicians, all very well intentioned. So this is not a case of people being daft. This is something more interesting and more, in my eyes, structural. So I wanted to try and understand how all of these wonderful people coming together, working together for like a good cause, could end up with this really problematic selective pressure in hypothesis building of iterations of a pre-existing exciting theory, and then the neglect of more plausible or likely ones. Now, to understand this, I, as you know, went to look at how information was being generated, was being interpreted, and was then being shared, because I thought that is the way to try and understand how a hypothesis or narrative has come together. As I said before, you can see at the very top, at the start of the outbreak, you have the pediatrician and the house officers in those green dots. And then as you come down, the virologist is in the red. And you can see people sort of talking and sharing information when those circles are touching. Now, I think the two most obvious structural features, just looking at that straight away, are that most of the information was shared in this sort of dyadic interaction, so just two people. And actually most of it was done by phone calls, text messages, and emails. There's an exception to this, which is the other key feature, which is you can see maybe halfway down, there are these two bits where there's several circles touching each other. Now, the first, sorry, let's see if I can, anyway, I'm blocking it, but further down, there is also another one of these. Now, the first one is that presentation that that field epidemiology team gave, where they presented their findings to all the sort of heads of the field epidemiology team, Ghana Health Service, this specialist laboratory. And the second one, oh gosh, why is this playing up so badly? Oh, anyway, which you might not be able to see right now and is at the bottom, is from an email chain that was when an American researcher later on tried to pull together a lot of researchers in Britain and the US to investigate the outbreak, having heard about it. This will become particularly important later. Something that you can't see looking at that, but I know, having looked at the documents, is that most of the people on that list were recruited to the response by a particular story. And that's really important for trying to understand where things went wrong. So that doctor was initially brought over because something was unusual. And then the doctor contacted the district officer and kind of recruited him to the response by the idea that it was a viral encephalitis, and a novel or unusual one at that. Now, he recruited the virologist on the grounds that it was a suspected viral encephalitis outbreak. And I mean, you can already see a slight problem here, because then the virologist only ran a panel looking for viruses that cause encephalitis. So any false positive you got there was going to simultaneously confirm the clinical impression and the laboratory results. Anyway, sorry, that's by the side slightly. But then once we had that positive suggesting B virus, the field epidemiology team was recruited to the idea of a laboratory confirmed B virus outbreak that fit with the clinical picture of viral encephalitis. You have this kind of snowballing confirmation bias. There is actually a somewhat larger problem there. And it's normally at this point in the talk that I talk about cognitive biases too. So for any other field epidemiologists in the audience, we are really susceptible to something called anchoring bias, which is the first piece of information we're given about an outbreak colors the way we interpret all subsequent information, and also bandwagon effect, which means the more people that are already talking about a narrative or committed or somehow associated to it, the more kind of credence we give it. Anyway, I just thought I'd mention that in case there are some out there. Policemen and investigators and detectives seem to be more aware of these issues, whereas field epidemiologists less so. Now, the bigger problem with the recruiting of responders by the story, rather than them being some generic investigative sort of structure, like the district officer and the pediatrician, is that they only really have resources, expertise, and interest in their area in that particular area. So, I mean, as long as it's something to do with a novel virus, that virologist has something to sort of contribute, share, and move forward with. The senior Ministry of Health officials, if it's something unusual, not just kind of a normal handful of malaria cases and such, that's something for them to keep pushing resources into and pursuing. The naval officer, this American researcher that later came in and very interested in this response, started to start an investigation and such of his own. Again, it was, I mean, they're not really going to do this for just another 16 cerebral malaria cases. It probably has to be virological and it has to be something interesting. So the problem is that when you have alternatives that aren't that this is a virus, say a potentially toxic etiology, most of the people that have sort of come around this outbreak aren't able to really perpetuate that, move that forward at all. Now, this should not really be a big issue, because in theory, you have your district disease control officer, who is meant to be the one that's sort of running the response and such. And I've just realized that I've got some things. All right, sorry, bear with me, COVID brain. I'm going to go from there and just actually illustrate what I mean by people that are recruited to the story are able to move hypotheses relating to the hypothesis they were originally fed forward, but not alternative. Bear with me. Okay, so going back to that forest belt, which definitely doesn't exist, but supposedly has the monkeys that make all of this sort of outbreak make sense. That first came about during that field epidemiology team's investigation. And it came about because one of the 3 families they interviewed, the father of one of these affected children, said that back in the day, and remember, they went out there looking for monkeys more or less. It was like, back in the day, I did once see a monkey at the bottom of my farm. And there happened to be a few trees in that particular village. So as the district officer who went with the team said, at that community we visited, we were told there was a forest around the community where they had found monkeys in the forest. Okay, so be it. That then became, in the minutes for the field epidemiology team's presentation, all affected districts were surrounded by a natural forest monkey belt. And that eventually became in that American naval researcher's proposal, that the source likely emanated from human-monkey interactions within a known forest monkey belt, which then became a research proposal that talked about the surrounding forest of all the affected districts having monkeys. And that eventually became a draft publication that also mentioned this natural forest belt. And it was actually edited by the pediatrician who made the point that the monkey mentioned in the original draft wasn't in the area, so it had to be one of these other kinds. So as you can see, non-existent monkey belt, it spreads freely and across different actors. This is a pet peeve of mine, and it's not just a pet peeve of mine. I want to make a point here too, that this shows that a lot of the people who were building off each other's information were unaware they were building off each other's information. So that American naval officer researcher that took the initiative later on and incorporated this forest belt into all of his work, he never spoke to the field epidemiology team that initially came up with this idea of a forest belt. He wasn't even really cognizant of them or their investigation. And yet it became such a key part of his later narrative. Now, why this is important to me is quite often, especially foreigners, come in for outbreak investigations, and they're just so oblivious to the work and the data generated, bad and good, that goes into the the construction of the hypothesis, all the baseline data that's needed, all the laboratory results, all the working with the patients and case finding and such. And I thought this was quite a nice case for that. Anyway, moving on. Hypothesis building. So we've seen that the invisible forest belt spreads quite easily, but when it became things that challenged the idea of a virus causing this outbreak, or posited an alternative theory like toxic etiology, as we can see, it doesn't go anywhere. So it comes up in a conversation between two people, and then it's not spread anywhere else. It's not mentioned anywhere else. So that top one is a medical student talking to the pediatrician, because the pediatrician, when they got the B virus results, was like, can you quickly do me a literature review on B virus, because I've never encountered it before, and it's obviously very important. And this medical student wrote, honestly, I think the chance of a false positive, an a priori false positive, is much higher than this being B virus. But that didn't really go anywhere. And later on in that field epidemiology team presentation, senior officials questioned how a 6-month-old baby was left unattended on a farm to interact with monkeys to contract this, and also why none of the children had bites or scratches or any of the other obvious ways that B virus typically gets into humans. In fact, almost all the B virus cases known, again from East Asia where there are macaques and such, come about because of lab accidents or mishandling of caged monkeys, or once cleaning the skull of a dead macaque. It normally takes a cut and a bite, and it's quite dramatic. And then later on, some British vets that were interacting with the American officer noted that they thought it looked toxic. But that conversation never really went any further, and no one really explored it. And in fairness, there aren't really that much in the way of resources to do a toxicological investigation. So we can see where the selective pressure, because of the people involved, is beginning to come from. Now, this should not be an issue because of that district officer, that sort of generic public health official who only cares about seeing the local population get well again and hasn't really got a horse in the race in terms of etiology. But something had happened in these kind of more senior figures coming in, which is that there was a substantial gradient in terms of authority and resources. So to give you a sense of this, the district officer had really wanted to do more case finding because he suspected that there were more cases in other villages. It was really the ones that had roads that could get to the hospital that had, presented children. But he wasn't able to get the money enough for fuel for his motorbike to do case finding or to print some case investigation forms on paper. Whereas the research proposal that came about with the American Naval Group was for $100,000. And again, to put this in context, the district officer just needed 60 Ghana cedi, so like $20, to do some actual case finding. But there was something more profound that had happened, which is this district officer had been displaced from the hypothesis building, from the narrative making. Those two events, no, it's not going to work. All right, those two events where the kind of narrative was built and everyone came together and made this sort of authoritative account, the district officer wasn't invited to. He wasn't invited to the field epidemiology team's big presentation with the national heads where they made the decisions going forward. And then he wasn't part of the sort of research proposals later on with all the American British researchers and the pediatrician and the virologist. He was just sort of cut out of that step. This is problematic, obviously, because it gets in the way of being able to explore other etiologies, but there's more to it than that. It disrupts the whole bringing in of information, the testing of hypotheses, and the ability to make new hypotheses to explore. Does this through two ways, the first of which is called variable dysfunction of information. Now, if the district officer had been allowed to be the sort of center of the response, or the sort of the central factor, then the information would have flowed into him. So he should have got the lab results, he should have got the clinical bits, he should have got all the pieces together. But as we can see from the spread of information, that's not what happened. It just kind of spreads between people and all over the place. And very rarely does someone actually get the information from the person who generated it. This combines very, very poorly with something called peripheral uncertainty absorption, which happens a lot with experts, where they kind of smooth information. So the virologist made no secret of the fact that the test was this experimental, unvalidated PCR and such. But there's only so much of this detail that he could communicate then on to the next person. So then it became, on this PCR, I got a positive for B virus rather than all the normal caveats. And eventually that to the FETP became laboratory confirmation. And the same happened with the clinical picture. The pediatrician knew that a chunk of the children had been afebrile. She knew that most of them had normal white blood cell counts. She knew that 4 of them had epistotonus. 2 had been administered acyclovir, which is a treatment for B virus, and it just so happened they survived. It was a bit of a confounding factor. But eventually that became 16 cases of viral encephalitis with fever, seizures, and improved outcomes with acyclovir. It gets kind of smooth like that and the uncertainty removed. Now, what happens when these 2 things combine is no one has the raw information and no one has a nice kind of dirty early account of the raw information either. Like no one has access to everything, which makes it very, very hard to then challenge this like B virus hypothesis, because you're like, oh, well, I guess they had the lab results and the clinical picture fitted, even if you know that the monkey bit isn't quite right. Whereas actually, if you'd seen all of it, you might have realized it was a bit messier than that to begin with. This is slightly separate from the rest, but I think it's important to mention here, which is the way that the bringing together of all these other people around this exciting, potentially high-profile outbreak changed the spatial and temporal aspects of the theory building, of the hypothesis building. So to give you a sense of it, the virologist, the senior public health officials, the naval officer, the other researchers, the British ones and such, they never visited any of the affected communities. Not one of them ended up in the Brong-Ahafo region, despite maybe 2 years of research proposals and such. The field epidemiology team spent 3 days in the affected area, and they managed to visit 3 of the 13 at the time affected communities. Now, the pediatrician and the district officer had both visited those 3 communities too, having lived there, but none of the others either. The area where the outbreak was going on and the people affected very, very rarely seen. Most of them weren't at all. This is obviously, it allows for some silly things like the forest belt to exist, because if you've never seen it doesn't exist, then that makes that easier. Same with the monkeys. It has another issue, which is it makes it very hard to do descriptive epidemiology properly. Descriptive epidemiology is where you're able to describe the geographic spread of cases, their sort of demographic feature, you do lots of case finding, you get a really sense of the time and the space. It takes a lot of time because you have to be there week after week, looking for cases, seeing if they've recovered, if they've died. It's the kind of ongoing, very much sort of, you had to be there type thing. Now, the third problem, which is the one that I'm maybe most interested in, especially when it comes to research about public health, is the abstract of urgency that can occur if you are not actually looking at someone who is suffering. This gets into this whole kind of messy area of pre-reflective ethics, which is the kind of ethical imperatives you have even before you sort of think about it. So when you are close to someone who's suffering, most people, I think, feel obliged, or compelled is maybe a better word, to intervene, to meaningfully stop them suffering. And sort of some kind of abstract version of that doesn't really cut it. You can't really do something symbolic really to help someone that in front of you is, say, injured or hurting. Now, I think when that's not there, that sort of proximity, and that proximity isn't just geographic, it can be terms of kinship. There's lots of studies showing that Black children in American ERs wait longer and are less likely to get pain meds and all these other kinds of things, especially if it's a white doctor. I mean, it can be a kinship difference, it can be a geographic distance, quite often it can even be a class distance. Anything that sort of gets in the way of this sort of, I guess, proximity ethic. With all these people not actually belonging to the affected community and also able to leave it, I feel like a lot of the urgency around the outbreak and a lot of the thing that normally drives people to do meaningful intervention was missing. So I mean, for over the course of, I think, 3 years, between September 2011 and March 2014, verbatim in all of the sort of research proposals for this outbreak and such that came out of that research coalition led by the American researcher. It said the immediate urgency for the prevention and treatment of these encephalitis cases. There was this urgency that just kept coming up in the reports, and yet no one actually bothered to do the 6-hour bus to the affected area to see if there were cases. I think it's also worth mentioning here that after those initial 16 cases kept showing up at the hospital. They just weren't really recorded because no one was really there. The doctor kept notes. So when I started looking at this, I think together we identified 52 suspected cases over the course of the years that followed. But it had all kind of dissipated by that. I think it's also worth mentioning that everybody involved in this, except for the district officer, has now left Ghana. If you belong to a community, I think it's a lot harder to let these things kind of go unattended to, or be able to sort of sign off on it without a kind of full conclusion. Whereas if you can walk away, you can kind of handle these more abstract summing ups of investigations, or ones that weren't like entirely satisfactorily described or resulted in public health intervention. The district officer kind of knew this was coming. So well before most of the investigations took place, he wrote this email to the pediatrician where he warned her, because some other people had shown interest in the outbreak, other foreign researchers, to be careful, because the little information people get on this disease, they want to publish because it's an emerging disease. However, there is a need for deeper investigation. Let's work as hard as possible to answer all the possible questions, irrespective of the years or months that it will take. They, the researchers, will come in, sit down, and they will take this one and this, and then they will come out with a nice story of a nice thing. I guess I might want to leave that as the kind of ending note for this investigation of hypothesis building and such, even though it's diverged slightly from hypothesis building, because I think it makes an important point about I guess, the larger point of internationalism in public health, and the fact that actually investing in core local health, public health infrastructures is maybe the only way to have meaningful investigations, but also sort of long-term health within a community. Now, I'm a little all over the place because this is very late for someone with long COVID, but I'm going to do my best now to sum up what happened with this outbreak and then move on to discussing COVID. So this outbreak, it's been years. It happened in late 2010. It's now late 2020, so about a decade there. It's hard to say for sure because the pediatrician who was the person that was keeping notes and kind of counting cases has left, but I think it's still going on. I don't really think that it was a B virus outbreak or any kind of simian herpes virus outbreak, and I think if I had to guess, I would say it was either an enterovirus or something toxic. But I think something happened. I think a public health emergency happened, and I think it's still happening. Now, the reason I think that's the case is because the annual polio data for Ghana, that area that was affected is not particularly populous, but it reports most of the area's suspected polio cases. And why that's interesting is because the way that suspected polio cases are counted are essentially unexplained paralysis in a child. So if a kid falls down with paralysis and there isn't much of an explanation, then this really exhaustive polio surveillance system in Ghana tends to capture them. And they send a sample to get tested for polio. Now, Ghana has not had any polio cases for a very, very, very long time. But this part of Ghana, the Brong-Ahafo region, still every year reports a large number of suspected cases that are then test negative for polio. It works out about 4 or 5 times the number of acute flaccid paralysis, so children suddenly developing paralysis, that you would expect for that area given the population. So in 2012, 102 cases of unexplained paralysis were reported. In 2014, about 97. In 2016, about 97. So that's a lot of unexplained paralysis. And I mean, you can find it every year in the polio data, but it never seems to raise a flag that this is a public health emergency going on. That's my main area of interest now. It's these obscured burdens of disease. So the sort of investigating hypothesis building and such, it's still very interesting to me. But my attention has now been drawn to trying to understand how you can have large burdens of disease, like this one that can just tick over in populations or in particular communities without generating some kind of excited public health response or intervention. I'm going to very quickly explain what happens now when you get a case of acute flaccid paralysis in this part of Ghana, because I think that that will help perhaps it'd be a nice sort of step into discussing this next bit about COVID. So if you have a case with acute flaccid paralysis in Ghana, a district officer is normally summoned. They're normally picked up by a little clinic or a hospital or such, and they fill out this form and they write down the details of the case and they pick up the sample and they send it to that polio reference laboratory. And like a lot of places, if the test is negative, they never hear anything back. If it's positive, they assume someone is going to call. So most of these forms for investigating acute flaccid paralysis, it just has a blank bit on the lab results. Though, as you can see, slightly further down, they do then follow up with the child a few months later to see if they've recovered or not. Then what happens is these forms sit on the desk of a district officer for a few weeks. And really what they're doing whilst it sits on that desk is they're waiting either to get a phone call saying it's polio, or they're waiting to see if suddenly there's a lot more cases, you know, to kind of tip the scales of goodness, we have an outbreak. But then when that doesn't happen, it just gets kind of filed away and forgotten. So that's a sort of, in the bottom right corner, you can see a storage facility of these forms. It's a way of, I guess, disappearing an aberrant public health event that doesn't fit well within the system and the constraints, material and otherwise, that people have to work within. And as such, this kind of stable burden of disease just kind of exists, and it's easy to find in graphs and such, but also just doesn't exist, and then it never generates action. Now, this brings me to COVID-19. So I thought that it would be nice to talk about long COVID. And the reason I thought it'd be nice to talk about long COVID is because I think it's a very good example of the sort of the processes by which a large burden of disease can become obfuscated. Now, the other reason, as I mentioned, I want to talk about long COVID is because I have never worked on outbreak investigations within the UK or any non-resource limited setting, except for maybe Australia. And that means that I feel very guilty trying to begin to talk in any kind of academic way about it. But as I've had long COVID, I think I can just about get away with calling this an autoethnography. And claiming that because I've lived the experience, I am allowed to now sort of comment on it despite not going through the formal academic steps. So for those that are not aware, long COVID is, I guess it's a sort of a manifestation of COVID that doesn't get better after 12 weeks. Oh, that's not quite right. It's essentially when people who got COVID and then 12 weeks later, they've still got some degree of symptoms, normally quite bad ones actually. Otherwise they tend not to bother giving it a label. It looks like it was mostly in people between the ages of 20 and 60, though it's still not quite clear. And a lot of them were previously fit, so none of the sort of comorbidities or cofactors that people talk about with sort of severe acute COVID. And it also looks like it doesn't really matter how sick you initially were with it, you can still develop this sort of chronic form of COVID. So some people got COVID very mildly for a week, but then months later, they're suffering a lot with these sort of ongoing neurological problems and shortness of breath and such. Now, as you've probably noticed, I'm not really just interested in outbreaks or burdens of disease for the disease they represent. I'm interested in what they tell us about our processes. So I thought I would quickly use long COVID as a case study to discuss where we can go wrong with case definitions and laboratory testing and maybe doctor-patient relationships and how this can feed into hiding burdens of disease within communities. I am fading fast though, so I'm not sure if I'm going to get through much of this. So these are the other outbreaks I'm going to draw on for this because I felt bad mentioning them in the original blurb for this talk and then I'm not actually discussing them at all. So case definitions. Case definitions, if you can remember that early slide of the steps of an outbreak investigation, a case definition is the sort of set of criteria used, standard set of criteria, to identify the cases that are associated with a particular, let's say, outbreak or burden of disease within a community. They tend to include just a few sort of key clinical features, maybe a date, maybe a geographic area, and sometimes a sort of lab result. So for an Ebola outbreak, the case definition you might want to use is a fever plus either vomiting and diarrhea, and maybe, though I doubt it, probably a negative malaria test. When you have an outbreak where you don't know what's causing it, it falls on the outbreak responders to kind of create their own case definition, which sounds very straightforward, but it's actually really, really tricky and gets us in trouble all the time. When you get a case definition wrong, it can really, really snowball, actually. You can end up giving an event, an outbreak, a sort of artificial geography by saying that to be counted as a case, you had to be within this area. You can give it an artificial sort of epicurve or timing, because if you say cases from November 2011, all of the previous cases kind of go missing, and you kind of give yourself a fake event start date. If you give the wrong clinical picture, you can end up missing most of your cases, or you can miss a particular subset of cases that have a slightly different clinical picture, or a very different clinical picture from the first ones you picked up. Almost all of these things happened with COVID. So, I should also say, in other outbreaks, a common problem, just in case there are some other field epis out there, is adding fever in automatically, just because most of the outbreaks you respond to are infectious diseases, you assume there's fever involved. There's 2 toxic outbreaks on that map. One was a heavy metal poisoning in West Africa. And in both of those cases, early on, I think just habitually, people added fever to the case definition, but as it was heavy metal poisoning, fever didn't really present with most of the cases, and they later had to go back and remove it to actually, find all the affected children. That's beside the point, going back to COVID. So with the COVID case definition, there are a lot of problems. So I think China, last time I checked, it got through about 7, and I think most people will probably be aware of one issue where they changed the case definition 1 week, and maybe not across the whole country, and suddenly there was a jump in the case number that seemed quite stark, and that was just a sort of surveillance thing. But ignoring that, the larger problems with the case definition is, I think early on, though I was struggling to find the source now, the case definition involved this seafood market, this wet market. Now, the reason why that was a bit of a problem, and I kind of noticed it was an issue, was that it obscures human-to-human transmission. So when you make a particular place part of your case definition, you can make it look like something called a point source outbreak. That's because when you have people presenting, say, with these respiratory tract infection symptoms, which could be anything at the time, if you only count the ones that were associated with this one area, then you miss a lot of the cases that came from other people. And I think that did happen early on in this outbreak. It did look like there wasn't human-to-human transmission. There was a suggestion there wasn't, because this was the way that cases were being found. Now, there was another issue, which is that typically, again, if you probably just hung around and you looked at the cases as naively as you possibly could, you'd get away with this. But people tend to sort of have default symptoms or ideas about an illness in mind. So the early case definition also went with sort of cough and fever. I mean, again, this happens a lot, and it's okay normally because you have this iterative process where you're like, oh, damn, take the fever out, or, oh, actually not that many people have coughs or something like that. And we did see that happen, but it can also snowball really badly, which we saw happen too, which is by April, there was this meta study on symptom prevalence. And in this meta study on symptom prevalence, it's had most cases with fever. It had most cases with this dry productive cough. I mean, this meta-study covered 24,000 cases of COVID across 148 different studies and 9 countries. And I'm so annoyed that I've managed to screw up that. There we go. But as you can see, something that we now think of in terms of COVID, a loss of smell and loss of taste, weren't really captured at all. Now, my theory is, up until April, people weren't really aware of this. So what happened was you have these case definitions that find particular cases and therefore over-report certain symptoms, and then they also ignore other ones. And so then you can get this situation where you have this great statistical or quantitative power, but actually it's not representative of a lot of the cases. Now, more recent symptom prevalence work has suggested that actually fatigue and headache for most adults and loss of smell are the most dominant symptoms. And actually cough and fever, not so much. Cough in maybe less than half of cases. Now in the over 65s, you do have more fever, so up to 41% of cases, but it's still a lot less. And below that, we have the ECDC, where they have loss of smell in maybe 70% of sort of mild, moderate cases, whereas I think fever in 45%. So we can see that's a very different clinical picture that eventually emerges. Now, why is this relevant to kind of obscuring this long COVID burden of disease? Well, I think it's beginning to come out now, especially with some of the earlier work from Wuhan in China, which is that the long COVID cases, so the people that ended up being sicker for longer, they were less likely to be hospitalized, and they were less likely to have pneumonia, and they were more likely to have neurological symptoms. And this will become relevant later. They were less likely to have antibodies. So this is quite a different clinical picture. Okay, so I'm gonna see how far I can push with this, but I think it's only gonna be a few more minutes. And especially as I think I've hit 60 minutes, which is going quickly into what testing did for this. Laboratory results for outbreak investigations. I mean, this is one of the cruelest bits, I think, of training to be an outbreak responder, is there is this step, I think it was maybe step 6 in the list I gave you before, which is build hypothesis, test hypothesis, get laboratory confirmation. It makes it seem like this very straightforward thing you can do that then confirms your hypothesis. Whereas actually in almost all of those outbreaks that I showed on that map before, there were huge problems trying to get any kind of lab results. I mean, there's big logistic issues. I mean, like, honestly, if you're looking at something that's got a toxic etiology, your chances of getting laboratory confirmation are negligible. But even with infectious diseases, trying to get the right samples, especially in a resource limited setting, into the right kind of packaging, and then find the relevant reference laboratory. And often you have to take the sample at a certain point in the illness for it to work. And then you allow for all the false positives and false negatives. So for one of the outbreaks on that list, it was an enterovirus 71 outbreak in Cambodia. And the reason it was investigated as an outbreak of unknown origin was that almost all of the cases had died, which is not normal for enterovirus 71. It's very, very unusual. But about 60 out of 60 children that presented to these hospitals had died of it. Initially, when they ran the tests, they did CSF samples and throat swabs. They got negatives, and it actually took a different lab and a very different kind of sample to be able to get a positive for that. And that's really, really common. False positives too. There's an outbreak in there where initially it was attributed to yellow fever because there were 4 false positives for yellow fever on a panel. Why this relates to long COVID though, and the obfuscation of a burden of disease, is because the early advice, at least in the UK, when there weren't that many tests, so this is a logistical issue, was that all the tests would be with the hospital. And if you were young and relatively well and didn't have a risk factor, if you could, you should just stay home and wait out the illness. The idea was that it would only last for about 2 weeks. You'd feel a bit rough, but you'd likely be okay. What this ended up meaning was a particular set of patients didn't have access to PCR tests when they could feasibly test positive by PCR. And it also meant that they weren't being counted in any of the kind of work on cases with doctors and researchers, which was mostly going on within hospital settings. You would have thought that maybe antibody tests could kind of counter this, but as we saw from the earlier studies, long COVID cases were less likely to have antibodies, but also a lot of the initial sensitivity and specificity testing for antibody tests was done on this cohort of hospitalized patients. So the Cochrane review of the antibody tests kind of notes this too, which is that for the 54 studies of antibody sensitivity specificity they did, 46 of them were just on hospitalized cases. And the hospitalized cases tend to have more antibodies, be much sicker, and tended to be male and older. So even if you were trying to sort of extrapolate, okay, so for antibody tests, say 20% of people are false negatives, actually it was probably more than that, which again, on a much larger statistical level, makes the problem of long COVID on later sort of serosurveys much smaller. You can see that maybe I'm getting a bit technical there. Anyway, what this led to is eventually was prevalence being undercounted dramatically when groups like the wonderful Zoe COVID app tried to estimate long COVID cases, but only counted people using the app who had tested positive, which again ends up skewing it substantially. So they ended up finding about 2% of COVID cases turn into long COVID, whereas studies from other countries put it much, much higher, about 10 to 20%. And I think one from the Faroe Islands today suggested 60% of people months on still have at least one symptom. I'm going to leave that there. I was really interested in looking at the doctor-patient relations, but I've exhausted time and I've exhausted myself. So I'll stop, and please throw questions, especially clarifications at me, and I'll do my best.
Dr. Jephcott, thank you for a fantastic talk. So now it's time for the Q&A session. So if you have any questions, feel free to ask them now. Okay, so Dr. Eisner?
Hello, not doctor, I'm afraid.
Okay.
Can you just clarify? Thank you for a fascinating talk. Can you clarify what PCR tests are?
Oh, sorry about PCR is when you're looking for evidence of actual virus. I mean, different context, different things. You're looking for evidence of actual virus, and what you do is you take a swab or a sample from someone, and then you put it through this process that makes the RNA or DNA that belongs to the virus much, much larger, to a point where you can actually pick it up, and you can, sorry, again, brain, you can actually identify what virus it belongs to. And so you can kind of say, oh, this virus infected that person. Sorry, this was a terrible explanation of PCR. Essentially, it looks for like RNA or DNA of a virus and tells you if it's there, if it works well.
Thank you.
Hi, I have a question. So the long COVID stuff, it reminds me a lot of CFSME, which is something I've had for 7 years. And the kind of quite common thing with CFSME is that it happens in 10% of people with glandular fever. But again, it's been something that's been entirely ignored by the medical community, to the point where it's actually just really kind of pretty criminal. And there are, I think, several reasons for this. Like, part of the reasons are that it's not so quantifiable in the kind of simple ways that are kind of easily accessible. And I was relating this to you saying how people with long COVID tend to not really show up on the antibody tests. And then, however, this then leads to people not really believing that there's a problem and you being invalidated, which is particularly key with chronic fatigue syndrome, because, and I think it's also very much a gendered thing, because I know the statistics are that 75% of people who develop chronic fatigue syndrome are women, and it tends to be young women as well. So I was also wondering whether there was a gender kind of bias on that as well, whether you knew about that. And yeah, whether you had any kind of advice to stop this kind of cycle of kind of not having something be immediately quantifiable count as evidence as something not being real, which then means there's no research into it. Then because there's no research available, it's obviously you're making it up, and it just goes into this awful, awful spiral where myself and pretty much everyone in chronic fatigue who's had chronic fatigue or experienced chronic fatigue or any kind of illness like fibromyalgia, and I'm sure long COVID as well now too, is just completely invalidated by the medical system over and over again, and by science as well.
Yeah, absolutely. So if I'd kept going, I was actually going to look at the doctor-patient relations and that side of it. I mean, it's not just like fibromyalgia and such. I mean, multiple sclerosis was hysterical paralysis until MRIs came about. Yeah. Epilepsy was hysteria too. I mean, MS and such, obviously a lot more prevalent in women, as most autoimmune disorders are. I mean, even stomach ulcers up until Helio pylori was identified as the bacteria that causes it, people were just like, stomach ulcers are caused by stress. Western biomedicine has an obsession with psychosomatic illness being the explanation of things that they can't easily image or test for. I think it's more than that. So in general, with outbreaks, and when people are able to make an outbreak obscure or a large burden, in my experience so far, it always had to be marginalized groups. So it was typically an area of Vietnam that's quite remote in an ethnic minority, or it was in disabled people, like people in dementia. A virus could sweep through, kill a lot of people, and it would take years to notice. Or, I mean, rural areas, poor areas, that's where you can make these outbreaks, these difficult situations kind of go away. So I thought long COVID wouldn't have such a problem because there's so many of us suddenly, like really, if you can get a sharp epicurve, that tends to get more attention. So the fact that there was suddenly like, so the Facebook support group for UK sufferers that I'm part of has, I think, 30,000 people in it now. And I'm friends with 2 doctors with it and another person from the disease dynamics unit with it. So I was like, it's very prevalent. It's affecting men and women. And there are so many doctors that have it. So I was like, okay, that actually bodes well for it. I was chatting on Facebook to a male doctor friend from a hospital the other day whose GP wouldn't refer him to a long COVID clinic because he hadn't had a positive test. And I was a bit stunned that if it's that bad, that maybe there's more to it than this. I'm really looking forward into digging a little deeper into the habitual and cultural practices of disappearing epidemics in non-resource limited settings like the UK's. Because Emmy, the whole situation, I was blissfully unaware of it up until recently.
It's diabolical.
It's horrendous.
The way the
absolutely the gaslighting of patients in this way, and the neglect in terms of research funding is really appalling. Hopefully, long COVID might. I don't know. I hope that somehow it lends
it some credibility.
Credibility and just like much needed research support and treatment. Yeah, I'm so sorry that you've been.
Because at the worst end of the spectrum, people actually die because they cannot digest food anymore because they are that exhausted, their body is that drained, and it's still seen to be psychological by the most prominent psychiatrists in the UK. I think Michael Stark or something, and they have, because it's their specific, sorry, I'm going to get on a rant now, but it's their paradigm, and there's so much kind of nepotism and
I'm hoping there's going to be a reckoning at some point soon. And I'm definitely, now that I'm aware of this, I'm fascinated by it, but also appalled. So I'm going to try and do some research into this too. I mean, ignoring large burdens of disease has massive consequences for society generally, as well as being unethical. So yeah, sorry.
Well, thank you for your thoughts. I'll be interested to see in what happens with the general public's response and the general medical system's response to long COVID patients.
Yeah, I'm not crazy hopeful, but we'll see.
Are there any other questions?
Is there anything in the chat, perhaps? I'm a bit shy, so I didn't put my questions there.
Yeah. Yeah, there is a question. What is the most robust way to estimate long COVID prevalence?
Oh, okay. So I think the way I would probably do it, though it would take a bit of sort of sample. I mean, it's a bit tricky because they are doing the testing within the UK is quite fractured. I think I would either try and piggyback on one of the big research projects that's already randomly sort of sending out sampling tests to get sort of PCR tests generally within, I guess as the outbreak goes down. Anyway, so I would piggyback onto some of the research already being done, just doing PCR tests on the population at large, so those big testing things, and then follow up with people who had positives, everybody, 3 months later and 6 months later, and ask them if they're well yet. I think that's the most obvious way to do that and what symptoms they still have. Otherwise, I think that maybe some of those things like the Zoe app could be used to come up with a reliable estimate if they're careful. Yeah, I mean, it should be straightforward. I'm kind of annoyed that it hasn't been done, but again, people tend to lean towards the more easily quantifiable, like deaths and such, or hospitalizations.
Okay, then there's another question. This is a bit longer. How do you think these uncertainties in diagnosis and detections could affect epidemic forecasts? In other words, how should modelers take these factors into account when they are trying to inform management strategies and policymakers?
Beware of your data. So I think before all modeling and all of that, it really helps to have really grounded, sort of detailed ethnographic and empirical accounts of the thing that the data is meant to represent before you go from there. And I think also there's been a wonderful growth in quantitative literacy, but I think we also need to bring up qualitative literacy as well, so that people developing models and such can engage with the social science research on the same topic and understand how they intersect and can also breed more accurate or more useful models. So I would say it would terrify a lot of people who work with public health data, how their data is generated. I mean, I did some research looking at facility-based surveillance for emerging infectious diseases in West Africa, and most people just get labeled malaria, despite whatever the cause was, simply because that was something that there was treatment available for. So the doctor only really wants to diagnose things they could potentially treat. And the fact that there just weren't really diagnostics for anything else. And then people will tell you, oh, there's this much malaria within this country, when that's probably not actually true. So yeah, just a lot of research into the countries or areas you're drawing data from. So again, a bit scattered.
Any other questions?
Hi, Dr. Jephcott. That was a fascinating talk. I'm really fascinated by the chain of events and how information gets changed throughout the process. And I guess I'm curious to hear what what's your personal perception of the reasons that these behaviors come out. Willingness to work with incomplete information or uncertainties, or omitting context. Do you think it's altruistic? People are just trying to do something good and they're willing to work with limited information, or do you think that there are selfish motivations, like the district officer mentioned in his email, or is it a lack of rigor in the field, or what's your take? Is there anything common, or is it all just a mishmash of human complexity?
So I think it's a mishmash. I would say that what I found most productive in trying to do my research is to assume everyone was generally well-intentioned and fairly able. From that, it's always such a mess of uncertainty. Like, I mean, that stuff was dodgy, but even the correct stuff is dodgy and blurry and ambiguous. That first step of verifying an outbreak is so much trickier than anyone could possibly realize. Just working out if something abnormal is actually happening can be really, really challenging. I think the thing that throws it most of the time when you have this kind of structural issue where information is going all over the place is just we naturally smooth things into a coherent story. We don't relay kind of disparate, confusing, disjointed bits of information to each other. We tend to say, oh, this kind of nice monkey-born outbreak with a forest kind of thing, rather than, yes, 4 cases of low white blood cell count, that kind of thing. It was a bad answer, but maybe I can talk to you about it later.
That's great. Yeah, thanks so much.
Are there any more questions? Yeah, probably not. So, Dr. Jephcott, many thanks for the great talk and for answering the questions. And this Thursday, there will be a talk by Dr. Matthew Wingate, fellow at Fitzwilliam College and a reader at the Department of Applied Mathematics and Theoretical Physics in Cambridge. And Dr. Wingate will be talking about his research in quantum chromodynamics. And the talk is going to start at the usual time at 6:15. So thank you for attending the talk and have a nice evening.
Thank you.
Thank you.
Thank you. Thanks.
Fascinating talk. Thank you.